So welcome back for part 2 of chapter 3. In this lecture, we're going to cover the remaining macromolecules, the nucleic acids and the proteins. So the nucleic acids are basically your DNA and RNA. There's another nucleic acid known as ATP and GTP. Those two are part of metabolism, and we'll talk about those quite a bit in our second unit. But for now, we're going to talk about nucleic acids in terms of DNA and RNA. Nucleic acids are collectively responsible for our genetic information, so storage, expression, and how our genes are transmitted and translated into proteins and other functional molecules in the cell. There are two classes of nucleic acids, deoxyribonucleic acids, which make up DNA. That's your A, T, C, and G that you probably learned about in previous science courses. Then we have the ribonucleic acids, which are your RNA, and those consist of A, U, C, and G. So they replace the T with a U. DNA is responsible for storing genetic information. It occurs in long sequences, and some of these sequences are broken up into shorter sequences called genes, and these genes will be transcribed into RNA. The RNA is responsible for decoding the DNA and taking information from the DNA to ribosomes and other cellular structures to carry out specific functions in the cell. So we have the exact definition here on the slide. Deoxyribonucleotides, there are two basic classes, the purines and pyrimidines. Easy way to remember it, the purines are adenine and guanine, the A and G. And purines, I always think of the saying pure as gold. So purines, pure, are adenine as and guanine, gold. So pure as gold, and that way you automatically know that the other two, thymine and cytosine, are the pyrimidines. So you just got to remember the purines, pure as gold, and you can then easily figure out which are pyrimidines. Purines have two fused rings, okay, two, and pyrimidines are a single ring. Now, when creating a DNA molecule, we know that it's a double helix. We have two strands of these deoxyribonucleotides in specific order, and they face each other, and they hydrogen bond in the middle and form that double helix that we're used to seeing. But when our bases base pair, when we have base pairing between adenine and guanine and thymine and cytosine, a purine will always bind to a pyrimidine. We never have a purine binding to a purine. That would be a mutation. So a purine will bind to a pyrimidine. In this case, adenine always binds with thymine, and it does so with two hydrogen bonds. Guanine will always bind with cytosine, and it does so with three hydrogen bonds. The monomer of DNA and RNA actually is a nucleotide, and the nucleotide is made up of a phosphate group attached to a 5-carbon ring sugar. That sugar is either a ribose sugar in RNA or a deoxyribose sugar in DNA. On the other side of this ring is going to be the pyrimidine or purine. So it's a single ring, it's a pyrimidine. If it's a double ring, it's a purine. These are carbon and nitrogen atoms, and they are referred to as the nitrogenous base. When nucleotides are bound together at the deoxyribose level, at the sugar level, when the sugar rings are bound together, they create what's called a sugar phosphate backbone, because the sugar ring is going to be bound to a carbon that's coming off of the phosphate groups. And they are bound together by a phosphodiester bond, which is a form of polar covalent bond. So here we have an example of a ribonucleotide, and down here is a deoxyribonucleotide, and you see the only difference here is we're missing an oxygen in the DNA nucleotide. But here is our, just highlight as we go along here, this is our sugar ring right here. That's the sugar base. The sugar ring. And here is our phosphate group, just sticking off the side here, and you can see there's a carbon in between here. And then coming off of the other carbon on the other side, opposite side of our sugar ring, is our nitrogenous base. So since this is a ribonucleotide, they used uracil. This is what uracil looks like. A deoxyribonucleotide is the exact same thing. The only difference is which nitrogenous base is bound to it. In this case, it's cytosine, and it's missing that oxygen. Otherwise, it's the exact same molecule. It's very similar. When these are bound together, I want you to notice something. In organic chemistry, we number the carbons. And because we have a molecule that has multiple rings and multiple carbon structures, you can see here we have our purines and pyrimidines. We number them a little bit differently. So the sugar, the base, the functional group is called the prime. So here we have carbon 1 prime, carbon 2 prime, carbon 3 prime, 4 prime, and here we have 5 prime. Our base over here would just simply be a 1, a 2, a 3, a 4, a 5, etc. But the sugar is really what we're concerned about here. And this structure is very important. The numbering of the carbons on the sugar portion, on the deoxyribose sugar in DNA and in the ribose sugar in RNA is very important to the structure of a DNA molecule in its entirety and how these are put together. DNA molecules have what we call directionality, and we'll spend more time on this in Unit 3, but I want to bring it to your attention now so that when we get to Unit 3, you're more familiar with it. So here I have my deoxyribose sugar, and if you notice, I have a 3' carbon right here, and this 3' carbon is bound to the oxygen of this phosphate group. This phosphate group is bound to the 5' carbon of the next incoming nucleotide. So this nucleotide is coming in, then this one comes in, and this one comes in. This type of bonding relationship, here we have another one. The 3' of this same molecule is bound to the phosphate group, bound to the 5' carbon of the incoming. 3', 5'. And so the next molecule here, we have a free 3' carbon. So the next nucleotide coming in, its 5' carbon is going to face the 3'. And this phosphate group here is the bridge between the two. This bond is referred to as a phosphoester or phosphodiester bond. Normally, a nucleotide would actually have 3 phosphate groups attached to it. And when this bond is made, 2 of those phosphate groups are going to be released, releasing the energy required to create this bond here, okay, right here. Now, these bonds continue throughout the entire molecule, and they are covalent bonds. So this portion here, right, this red portion here of the DNA molecule is very strong. It's thousands upon thousands upon thousands of covalent bonds all stacked up on each other. And we know that covalent bonds are the strongest types of bonds, the strongest form of chemical bonds. These bases over here, you notice all of these hydrogens are all sticking out over here. And when the second strand, this is just a single strand here, when the other strand of DNA comes in complementary base pairs, it will do so by hydrogen bonding. And in order to do so, it's going to be in the opposite direction. So DNA molecules are a double helix, but they are referred to as antiparallel. Now, DNA versus RNA, they're very similar. We have in DNA, we have a deoxyribonucleic acid, and in RNA, it's a ribonucleic acid. The only difference is that missing oxygen. DNA uses the sugar deoxyribose, RNA uses ribose. As far as bases are concerned, there are 4 bases utilized by both types of genetic material. DNA uses A, T, C, and G. RNA uses A, U, C, and G. DNA is double-stranded, RNA is single-stranded. DNA occurs in one form, just a double-stranded double helix, and RNA can actually occur in multiple forms. RNA forms all kinds of different shapes, and it can also carry out multiple functions. So here we have a double helix. We have both strands of DNA, and if you notice, on this side over here are 3' to 5' carbon, right? So this is the 5' end, because this guy's 5' carbon would be sticking up here, But this guy's the opposite. His 5' carbon is sticking out here. There's his 3. And he's moving 3 to 5, 3 to 5, 3 to 5, etc. So this is the opposite strand, and if you notice that it is going in that direction, and this one is going in that direction. That makes these two molecules antiparallel. They are antiparallel, so their bases both face the center and come into contact with each other in order to form these very important hydrogen bonds. We have 3 hydrogen bonds between guanine and cytosine, and we have 2 hydrogen bonds between adenine and thymine. And when we get into Unit 3, we will see why it's important that there's some differences in these hydrogen bonds and the structural purpose that serves. Now, DNA individually, these hydrogen bonds are very easy to break. Remember, hydrogen bonds are very weak individually, but when you have millions and millions and millions of them all occurring at the same time in a single DNA molecule, it now becomes a very strong molecule. We have millions of hydrogen bonds holding together these 2 strands, and the 2 strands are each held together by covalent bonds, and that makes DNA a very, very sturdy and a really strong structural molecule. Now let's move on to proteins. Proteins are made up of carbon, oxygen, hydrogen, and nitrogen. There are oftentimes small amounts of a few other elements in there, for the most part sulfur, but really they're primarily just carbon, hydrogen, oxygen, and nitrogen. An amino acid is the monomer subunit of a protein. The amino acids all have a common functional group, with each of them having their own R group. Remember, functional group versus R group in molecular structure. There are 20 amino acids that are essential for life, and that is because all of our proteins in humans, our proteins are made up of 20 distinct amino acids, and they're all put together in all kinds of different combinations to create all of the different proteins that make our cells work. The side chain, that R group, is what determines structure and function of proteins, because those R groups all interact with each other when these proteins are made. So let's take a look at an amino acid here. So here's the general design. Here we have our amino group. So this is your amine. Over here we have a carboxyl group, and we have a carbon and a single hydrogen. So our carbon has its 4 bonds. Right here, this R group right here, that is what makes each amino acid unique. Okay? So that R group is going to be different from one amino acid to the next. That's why when we talk about amino acids as a monomer or as a general structure, we draw them with this structure here, with R up here. And R indicates that it's going to be some combination of its own. But the amino group, carboxyl group, those are always the same. And when amino acids are put together to form proteins, it's referred to as a peptide bond. So here are different classes of our amino acids. We have polar and nonpolar amino acids. And in the polar amino acids, some of them have a charge to them and some of them are uncharged. And these properties of these amino acids, notice in yellow, they all have the same structure. Here's our amine group right here, and here's our carboxyl group right here, our hydrogen, and then here's our R groups, right? So this R group is a hydrogen, this is a methyl group. This here, over here, we have a whole bunch of carbons. Some of these are quite large. Here we have some ring structures. Tryptophan is a really big amino acid. So we have all of these R groups are different. Now, how these R groups affect the actual molecule itself, these are our nonpolars. And then over here, other R groups, depending on their structure, can affect others. And these are polar but uncharged. And here we have polar that are charged. In the charged, notice, we go even further and reclassify as acidic or basic. All of these different characteristics of all of these different amino acids, none of these characteristics, all of these characteristics of these amino acids play roles in how each of these amino acids interact with each other when a full protein is formed. You'll see what I mean when we get into protein structure. Now, proteins are put together by a dehydration or condensation reaction. The bond that's formed is called a peptide bond. When we have multiple amino acids put together, it forms what's referred to as a polypeptide. So a protein, the term protein and polypeptide are the same thing. Proteins can be made up of a single polypeptide, so a single chain of amino acids, or we can have multiple chains of amino acids that then also react together to form a polypeptide that's what we call quaternary protein that has multiple polypeptide chains all put together to create functions. We break up the peptide bond using hydrolysis. So whenever we have a condensation reaction where we're removing water, then if we want to break that molecule apart, all we have to do is put that water molecule back in. So here we have kind of the basic structure of how a protein is starting to be formed, or a polypeptide. So here's the peptide bond. The carboxyl group of one amino acid will react with the amine or amino group of the neighboring amino acid and form this peptide bond between the nitrogen of the amine group and the carbon of the carboxyl group. So in here, right, we have NH3, we lose 2 hydrogens, right? Let's say we lose these 2 hydrogens here. We lose this oxygen, and this carbon now binds to this nitrogen, and we end up with H2O. So that's a condensation reaction. This is referred to as a peptide bond. Because they line up, now notice what? If I were to bring in another amino acid, what do I have free? I've made this peptide bond, but now this carboxyl group, right? This carboxyl group is free. If I bring in a new amino acid, it's going to have an amine group that's free, and they can react with each other. So we end up with a peptide bond here, peptide bond here, peptide bond here. And all of these peptide bonds, because the functional group is the same within all of the amino acids, but the R groups are different, we can have multiple amino acids creating this long polypeptide chain, but each amino acid has its own characteristic. And these R groups up here, these regions, can interact with each other depending on whether they're polar, non-polar, charged, acidic, basic, whatever characteristic it is that they have. Let's say that these two here, this was a charged amino acid, and this was a charged amino acid, but they had opposite charges. They would repel each other. If these two amino acids here repelled each other, that would cause the polypeptide here to bend and start moving kind of like that. Okay? It would cause it to bend. And when these guys react with each other, it's going to cause the polypeptide to start taking on a shape. Right? Now, if we have a long amino acid, I just want you to know for future classes, when you get into your your upper division biochemistry classes, and maybe even into your medical or graduate classes, when they talk about proteins, proteins are linear and they have 2 ends. They're not circular. And the end in which the amine group is free, that's called the N-terminus, right here. And the other end, where the carboxyl is free, is called the C-terminus. Each amino acid will be given a number, so they're numbered along the polypeptide. And oftentimes they'll say, tyrosine 123 from the N-terminus. So it means that you start if you're looking at the structure of this protein, you would start at the N-terminus and count in 123 amino acids, and you would get to that particular tyrosine. So the amino acids will be numbered, and then you have the N-terminus and C-terminus, and they are going to become very important in your upper division courses when you are trying to locate specific amino acids within a protein. Sorry about that. Now down here we have our polypeptide, and you can see they've just taken away the molecular structure, and here's our N-terminus. We've numbered each of these amino acids, and here's the C-terminus, right? So this is called an octapeptide because it contains 8 amino acids. So that brings us to protein structure, because how amino acids are put together to create polypeptides plays a really important role in the protein structure. And protein folding or protein structure occurs in 4 levels. The first level is known as primary structure. Now here on the left we have our primary structure. The primary structure, here's our N terminus, here's our C terminus down here, and then we have our amino acids. So here, this particular sequence of amino acids is resulting in an alpha helix, which we'll find in secondary structure. This amino acid sequence here is resulting in a structure known as a beta sheet. It all has to do with how these amino acids interact with each other. They're neighboring. Now, in primary structure, DNA are genes, right? Are sections of our genetic material that encode for a protein. Our DNA tells ribosomes which amino acid to put in which order. So ribosomes read RNA. RNA is a copy of our DNA. So when ribosomes read the RNA, the RNA tells them, okay, first you have to put a methionine, that's this MET, and then once you have a methionine there, we need to add a proline, then we need to add a tyrosine, etc., etc., etc. So the RNA is telling the ribosome what order to put these amino acids in to create the polypeptide. Once the polypeptide has been created, depending on the order, that's going to determine how these amino acids interact to create secondary structures. So that brings us to secondary stage of folding. In the secondary stage of folding, we are looking for repeating sequences, and these sequences result in very specific, unique secondary structures. Alpha helix and beta sheets are the two most common secondary structures. Alpha helices are used quite often in proteins that are spanning across membranes or found in hydrophobic or hydrophilic regions, those sorts of things. Alpha helices are very involved in proteins that act as enzymes and transcription factors and interact with DNA itself. So here's our primary structure. And essentially, the meaning of primary structure is just simply it's an amino acid sequence. It is that sequence, what order these are put together in, is determined by our genes found in our genetic material. And the bonds that form primary structure are strictly peptide bonds. So they are covalent peptide bonds. This is done through, and again, remember, this is a condensation reaction. So if I want to break down a protein and break off amino acids, I would do that through hydrolysis. In secondary structure, we start seeing these interactions between neighboring amino acids, and it causes this folding. Alpha helices and beta sheets are the result of this. We can have irregular or repeating patterns, but when we have these interactions between neighboring amino acids, this causes our polypeptide, instead of being a big long string, it causes it to start kind of curling up and bending and taking direction. When we get these kind of random like coiled regions, and even when we get alpha helices and beta sheets, this now brings amino acids that were far away from each other and would not have any type of interaction. It brings them together. So now our amino acids that are far away are now being brought closer together, and their R groups can then start interacting, and we start going from secondary structures into what's referred to as tertiary. In tertiary structure, our protein now starts to fold in all kinds of different directions, and it takes on a real three-dimensional shape. If a protein is composed of a single polypeptide, meaning its function, the entire protein in its entirety is a single polypeptide chain, then that protein is complete and functional in its tertiary structure and is referred to as a tertiary protein. But some proteins require more than one polypeptide chain. Hemoglobin, for example. Hemoglobin is made up of multiple polypeptide chains. It's made up of 4. So we have 4 polypeptide chains are created and fold to the tertiary level. And then once they're in the tertiary level, the 4 of them interact together and become what's called a quaternary structure. So in a quaternary structure, we have 2 or more polypeptides making up a protein in its entirety, so it becomes functional. These quaternary proteins are oftentimes referred to as multimeric, and this is because they can actually have multiple functions. So here we have our tertiary structure. Notice the secondary structures, right? Here's our alpha helix here. Here's another alpha helix. That's part of secondary folding. The ribbon itself is made up of primary, right? Primary structure. Then secondary structure took place, and it's no longer linear, but now this portion, right? This portion of our polypeptide is now in closer contact with this portion of our polypeptide, and they can actually start interacting. You see that bond there. Same thing is happening here. Here in tertiary structure, we get an additional type of reaction called a disulfide bond. Disulfide bonds are very strong. And they can only occur when we have two sulfur groups that are close to each other. That's usually your cysteine, and I think it's methionine have some sulfur in them. And cysteine forms these disulfide bonds. But if this polypeptide was in its linear form, just a straight long chain, this amino acid and this amino acid would not be close enough to form this disulfide bridge. So here in tertiary structure, we're starting to see some real three-dimensional folding and shaping going on so that this protein can become functional. Look at how all these R groups are facing in the middle. Probably has something to do with the function of this particular protein. This is just another way of depicting. So we have our primary structure here. That leads to these secondary structures. We saw this before. When we go from secondary structures, remember these are now closer together, we move over here into our tertiary structure. And this protein is really starting to take on an interesting shape. Now, if a multiple polypeptides were required over here, right, here's our purple and aqua colored polypeptide that we created here, and it is now interacting with a dark blue protein that was created, and the two of them together create a quaternary structure. So there are 5 important factors to remember that promote the ability of a protein to fold and its stability. Proteins are pretty stable molecules, particularly our enzymes. Enzymes need to be stable because they work as catalysts, so they get used over and over and over again. So 5 factors that promote protein folding. Number 1 is hydrogen bonds. Lots of hydrogen bonds occur between neighboring and distant amino acids in both secondary and tertiary folding. Ionic bonds are also important and also occur between neighboring and distant amino acids in both secondary and tertiary folding. Hydrophobic effects, that would be a hydrophobic and hydrophilic amino acid, or we would have a bunch of hydrophobic amino acids all in one single region of a polypeptide because that protein's function is going to require it to be in a hydrophobic area. So we need those amino acids to be hydrophobic as well. If they were hydrophilic, they might be repelled by other molecules depending on the protein's function. Van der Waals forces play a really big role in protein folding. They're not, we talked about them a little bit in the beginning when we talked about types of bonds in chapter 2. I mentioned van der Waals. Van der Waals play a lot of important roles in folding and in how enzymes work. So van der Waals forces are that kind of 4th type of bonding. And last are the disulfide bridges. Disulfide bridges only occur in tertiary and quaternary structure. We do not have any disulfide bridges occurring in secondary structure. Protein to protein interactions are when we get those multiple proteins interacting with each other. Many processes involve lots of steps in which 2 or more proteins have to actually interact with each other. Metabolism would be one of those. This requires specific types of binding on the surface of each of these proteins. So these protein-to-protein interactions will use the first 4 factors of protein folding: hydrogen bonds, ionic bonds, hydrophobic effects, and van der Waals forces. Once in a while, we do see disulfide bridges occurring in these quaternary structures, like when antibodies are put together. Antibodies are proteins that are quaternary, and they are oftentimes held together by these disulfide bridges. So it's not particularly common, but it does occur. So here we have polypeptide 1 in its tertiary state. Here's polypeptide 2 in its tertiary state, and these two are designed, now by design, are meant to interact with each other to come together and form a quaternary protein. So these quaternary proteins oftentimes form what are called functional domains. And this can happen in a tertiary structure as well, but these proteins have multiple regions, multiple areas on one molecule that have their structure is what causes them to have very distinct function. Proteins are structure determines function. So your DNA determines the structure of the protein, and the structure of the protein is what determines its actual function. So indirectly, DNA determines the function of a protein. What's called a module or domain in a particular protein will have its own structure and its own function. We are going to just take a look real quick at an example of a quaternary protein or a multifunctional protein called STAT. STAT is a signal transducer and activator of transcription. This is a protein that goes into and interacts with DNA to turn on and off the transcription of different genes. So the STAT protein is important. Each domain of the protein has its own distinct function, and it actually has multiple domains. There are some other proteins out there that share some of these domains, and that domain will have the same function, but there are other parts of that protein that make it that carry out different functions. So the domain function is actually dictated by the primary sequence of the protein, and this is a little easier to understand if we take a look at this particular protein. So here we have our STAT protein up top, and the colors correspond to the colored portion up here. So here we have what's known as the protein bonding domain. This domain here, this region, this polypeptide, allows this region of the protein to interact with other proteins in order to be activated or to activate something else. It serves as a site that can turn the actual STAT protein itself on or off. Here in blue is the coiled coil domain. This domain is what regulates how STAT interacts with other proteins in the nucleus of our cells. So this one is particular to nuclear proteins, usually particular transcription factors. And we haven't done gene regulation yet, so if some of this is a little confusing, I understand. I really just want you to get the concept that we have different regions of a protein, of a single protein made up of multiple polypeptides, and together, once they're all together, we have this really big protein that has multiple functions. Now in red, we have the DNA binding domain. So this is the region of the STAT protein that will actually interact with DNA itself. The SH2 domain is required for binding to other molecules to either increase or decrease activity. The transactivation domain, this purple region here, is required in order to activate very specific genes. So this portion here would make it very specific. And then coming off of that transactivation domain is our C-terminus tail. So here's the N-terminus, and all of these proteins, this is all one protein, but each of these regions, because they are folded and structurally different, each have their own unique function. But if you notice, all of these functions are related. They're all related to the regulation of genes. They're all related to the activation, turning on, turning off, interacting with proteins in the nucleus. All of these different functions of this single protein with multiple domains, they all relate to each other. You wouldn't have a protein, a STAT protein, with a domain that interacts in metabolism, because this protein is found in the nucleus and metabolism is not occurring in the nucleus. It would make no sense. So it's kind of common sense that you would have all of these different domains all play not similar roles, but roles that are related to each other and to the final function, which is regulation of gene activity, turning our genes on or off. So that is nucleotides and proteins, and how the structures where these macromolecules are coming from, and how the structure of a macromolecule really lends to the function of it. Some things you're going to want to know for your exam. You're going to want to know all of the 4 macromolecules. You'll need to know the 3 monomer units of 3 of those macromolecules. You'll need to know where hormones come from. You'll need to know the types of bonds that are used to form glycosidic bonds, peptide bonds, the phosphoester bonds, all the different types of covalent bonds that are formed between monomer units of the macromolecules. So is it a glycosidic, a peptide, a phosphoester, and is it a condensation type reaction? You will also need to know how to break apart these molecules, so hydrolysis reactions. You must know all of the details of protein folding. That is going to come back in other units and also in other classes. So it's very important that you're able to distinguish the 4 levels of protein folding, the types of bonds that are occurring in each of those, and the differences between tertiary, quaternary structure of proteins and the different reactions that occur in each. You also should be familiar with multimeric proteins, what multiple domains are. The STAT protein, I would definitely take a look at that in your textbook and read up on it a little bit. You might see that as an example in an exam. So that is all of chapter 3, and we will move on to chapter 4 next.